Hi, my name is Joanna Kalovis, and my URAF project focuses on curating a collection of seed bacterial community members that are enriched under maternal plant stress. Plants, like us, have members of their microbial community, and these members can help them to gain nutrients, fight infection, and withstand environmental stress. They can get these bacterial members of their community both vertically and horizontally. Vertical transmission happens when plants inherited their microbes from their parent, and horizontal transmission happens when they acquire these microbes from the environment. Seed microbes are important because they're hypothesized to aid in plant resilience to environmental stressors, and the ability to determine what effects specific microbes have on plant resilience to stress could really provide new methods for improving plant health. In figure 1, you'll see what I was just talking about, this methods of colonization. So on the left, you see an adult plant with multicolored microbes that then gives them to the seed. The seed also gets microbes from the soil, these dark colored shapes. And as the plant grows, you can see it has both horizontally and vertically transmitted microbes. Above that, you see a cross section of the seed, which is where you can find different microbial members. Some of our goals for this project are using the common bean, a staple crop, to study the effects of these non-living stresses, abiotic stresses, on the effects of the inherited microbial community. We want to develop a protocol to isolate and culture members of the seed microbiome from these plants that have been stressed, and we want to be able to add these members back into the plant to determine their effects on plant growth during stress. In figure 2, you can see how we applied stress to these plants. We took our generation 0 seeds and grew them up as our generation plants. We applied our different treatments, control, excess nutrient, and drought. And once the plants were grown to maturity, we were able to collect the seeds. And from those seeds, we'll be able to culture a collection of bacterial isolates. In figure 3 below, you'll see some preliminary data on the relative abundance of seed microbes. What you're seeing is each plant and the average relative abundance of the microbes. So the average amount of expected microbes and microbes that we saw and what class they belong to. And we use this preliminary data to inform the choices we were going to use for our media to grow these microbes. Our final choices included TSA, Jensen's media, R2A, and RIM. Unfortunately, there are some challenges to isolating these microbial members, and one of those is that seeds are notoriously low microbial biomass systems, so it's difficult to find the microbes in the seeds. When bean seeds are grown or activated, they release antimicrobials, which kill the microbial members we're trying to isolate. And a lot of the current methods to extract microbes from the seed use sequencing methods to determine the identity of microbial members, and we actually need to grow these members in the lab. So in table 2, you'll see different growth temperatures and additives that we tried to use to isolate these microbes, and something we found that didn't work was actually tween 20, which is a cell lysis agent. In figure 4, you can see some serial dilutions that were performed, and these were performed to determine the concentration of bacteria present in the sample. Finally, our next steps. We want to continue to develop a working protocol to isolate microbial members of the inherited seed microbiome. Then we want to be able to apply the protocol to these seeds produced from our treated G1 plants to identify and cultivate a representative isolate collection. Finally, we want to add these microbes back to future plants to determine what effect these isolates have to protect plants from abiotic stress. In the future, adding microbes to the seeds may be an important mechanism to increase plant tolerance and resilience to environmental stress. Thank you.